Introduction. Welcome, dear students, to today's lecture on the intricate world of DNA replication and its fascinating mechanisms. In this comprehensive script, we will explore the complex processes that ensure faithful duplication of genetic material in every dividing cell. Get ready to delve deep into the realms of DNA replication, origin selection and activation, initiation factors, cell cycle regulation, replication fork dynamics, RNA priming, DNA polymerase accuracy, replication fork stalling and restart, replication clamp loaders, chromatin remodeling, DNA damage response, telomeres and telomerase regulation, replication timing, and epigenetic regulation. Finally, we will synthesize all the learnings into a comprehensive conclusion. So, without further ado, let's embark on this captivating journey through the mechanisms that drive DNA replication. DNA replication is initiated at specific sites in the genome known as replication origins. The selection and activation of these origins are critical processes that ensure the accuracy and efficiency of DNA replication. Throughout the genome, potential origins are present, but only a subset is chosen for replication initiation during each cell cycle. These origins are marked by distinctive chromatin features and binding of various proteins, such as origin recognition complex, ORC, CDC6, and CDT1. Together, these proteins form a pre-replication complex, pre-RC, that licenses the origins for replication. The assembly and activation of the pre-RC are tightly regulated by cellular signaling pathways and ensure that DNA replication occurs only once per cell cycle from each origin. The precise mechanisms governing origin selection and activation are still under intense investigation and subject to ongoing research in the field of molecular biology. During the G1 phase of the cell cycle, the assembly of pre-RC involves a series of orchestrated events orchestrated by initiation factors. These factors, including ORC, CDC6, and CDT1, play crucial roles in licensing replication origins. ORC, the origin recognition complex, binds to specific DNA sequences within replication origins and serves as a scaffold for the assembly of other pre-RC components. CDC6 and CDT1 are proteins that interact with ORC and recruit additional factors necessary for the establishment of a functional replication fork. Once the pre-RC is fully assembled, the initiation factors are displaced, and the replication machinery can be loaded onto the origin to initiate DNA synthesis. The intricate interplay between initiation factors and other regulatory proteins ensures the precise and orderly initiation of DNA replication. The process of DNA replication is tightly regulated to ensure its accurate and timely completion. Cells have evolved exquisite mechanisms to synchronize DNA replication with the cell cycle. In the G1 phase, DNA replication origins are licensed for replication by the assembly of pre-RCs. Progression into the S phase triggers the activation of pre-RCs, resulting in the initiation of DNA synthesis. Checkpoint signaling pathways monitor the integrity of DNA replication and coordinate the cell cycle progression accordingly. These checkpoints ensure that DNA replication is completed before the cell enters mitosis and prevent the accumulation of genomic abnormalities. Complex networks of protein kinases, such as cyclin-dependent kinases, CDKs, and checkpoint proteins, such as ATM and ATR, govern the cell cycle regulation of DNA replication. The replication fork is a dynamic structure that forms during DNA replication and consists of two template strands and two newly synthesized daughter strands. The replicative DNA helicase unwinds the DNA double helix at the replication fork, creating single-stranded DNA templates for DNA synthesis. As the replication fork progresses, the leading strand is continuously synthesized using a process called continuous DNA synthesis. In contrast, the lagging strand is synthesized discontinuously in short fragments known as Okazaki fragments. Coordinated action of various enzymes, such as DNA polymerases, DNA helicases, and DNA ligases, ensures the smooth progression of the replication fork and the faithful synthesis of both DNA strands. During DNA synthesis, the priming of DNA strands with RNA molecules is an essential step. RNA primers provide a starting point for DNA polymerases to initiate DNA synthesis. Primus, a specialized RNA polymerase, synthesizes short RNA primers that are complementary to the DNA template strand. These RNA primers are then recognized by DNA polymerases, which extend the RNA primers with DNA nucleotides to generate new DNA strands. Priming of DNA synthesis by RNA molecules enables the semi-discontinuous replication of the lagging strand and ensures the faithful replication of the entire genome. DNA polymerases are remarkable enzymes that catalyze the synthesis of DNA strands during replication. These enzymes have evolved mechanisms to maintain high fidelity and accuracy during DNA synthesis. DNA polymerases possess a proofreading exonuclease activity that enables them to identify and correct errors made during DNA synthesis. Additionally, the incorporation of correct nucleotides is facilitated by the precise active site architecture of DNA polymerases. Certain DNA polymerases can also stall or disengage from the DNA template in response to DNA damage or other replication blocking events, ensuring the fidelity and accuracy of DNA replication. DNA replication is a highly coordinated process that can be disrupted by a variety of factors, such as DNA lesions, DNA binding proteins, and DNA secondary structures. When replication forks encounter obstacles, they can stall, leading to DNA damage and genome instability. Cells have evolved mechanisms to cope with replication fork stalling and ensure their restart to maintain genomic integrity. Specialized helicases, such as REC helicases, aid in the restart of stalled replication forks by unwinding secondary structures and resolving DNA-protein complexes. The careful orchestration of these mechanisms prevents stalling-induced replication fork collapse and DNA damage. To enhance replication efficiency and speed, DNA polymerases are tethered to the DNA template through processivity factors. Replication clamp loaders, such as the proliferating cell nuclear antigen, pCNA, complex, play a crucial role in loading processivity factors onto DNA strands. pCNA forms a ring-like structure around the DNA, acting as a sliding clamp that keeps DNA polymerases bound to the template for extended periods. This allows for rapid and processive DNA synthesis without frequent dissociation of polymerases from the DNA. The collaboration between replication clamp loaders and processivity factors ensures the high processivity and efficiency of DNA replication. The packaging of DNA into chromatin imposes an additional layer of complexity on DNA replication. During replication, chromatin structure needs to be dismantled and reassembled to allow for efficient DNA synthesis. Chromatin remodeling complexes, such as ATP-dependent chromatin remodelers, play a crucial role in this process. These complexes use the energy of ATP hydrolysis to reposition or evict nucleosomes, thereby facilitating DNA accessibility for the replication machinery. The coordination between replication factors and chromatin remodelers guarantees the faithful duplication of genetic material even in the context of the highly regulated chromatin structure. The accurate replication of DNA is constantly threatened by various types of DNA damage. Cells have evolved an elaborate DNA damage response pathway to detect and repair DNA lesions during replication. Checkpoint signaling pathways coordinate DNA repair processes with the cell cycle progression to ensure the fidelity of DNA replication. DNA damage sensors, such as ATM and ATR kinases, are activated upon DNA damage and initiate a cascade of signaling events that result in cell cycle arrest and activation of DNA repair machinery. The timely and precise execution of DNA damage response pathways prevents the propagation of replication errors and maintains genomic stability. Telomeres are specialized structures located at the ends of linear chromosomes that play a crucial role in maintaining genome integrity. During DNA replication, the replication machinery encounters difficulties in faithfully replicating the ends of linear chromosomes. To overcome this problem, telomeres are equipped with a unique enzyme called telomerase. Telomerase adds repetitive DNA sequences to the telomeres, extending their length and preventing the loss of genetic material with each round of replication. The regulation of telomerase activity is tightly controlled to ensure proper telomere maintenance and prevent uncontrolled cell proliferation. Aberrant telomerase regulation is associated with aging, cancer, and various genetic diseases. DNA replication is a highly coordinated process that is tightly linked to epigenetic regulation. Replication timing refers to the specific timing of DNA replication at different regions of the genome. The replication timing program is established during development and is regulated by epigenetic marks and chromatin structure. Late replicating regions are often associated with heterochromatin and repressed gene expression, while early replicating regions are enriched for active genes and euchromatin. The precise temporal regulation of DNA replication ensures the proper coordination of replication with other cellular processes, such as transcription and cell division. In conclusion, DNA replication is a crucial process that ensures the accurate transmission of genetic information in living organisms. It involves a complex system of factors and mechanisms to maintain genomic stability. Understanding DNA replication has implications for understanding diseases like cancer. As aspiring medical professionals, a strong understanding of DNA replication will help you comprehend the molecular mechanisms of diseases and develop effective treatments. We hope this video has provided you with a solid foundation for further exploration of DNA replication. Stay curious and enjoy the wonders of the molecular world.